Good morning. Today I am going to start the next topic, separation and isolation of the DNA fragments by gel electrophoresis. The cutting of DNA by restriction endonuclease results in the fragments of DNA which are separated by a technique known as gel electrophoresis. In another word, we can say that gel electrophoresis is a technique for separation of DNA fragments based on their size, in which first the sample DNA is cut into fragments by restriction endonuclease. The DNA fragments is negatively charged, can be separated by forcing them to move towards the anode under the electric field through a medium or matrix. The commonly used matrix is agarose. Agarose is a natural linear polymers of D-galactose and 3,6-N-hydro L-galactose. Which is extracted from seaweeds. The DNA fragments separate out or resolve out according to their size because of the sieving property of agarose gel. Hence, a smaller the size of fragments, the farther it moves. So, a smaller the fragments move farther. The separated DNA fragments are visualized after staining the DNA with ethidium bromide, followed by the exposure of ultraviolet radiations. The DNA fragments are seen as orange color band. Orange color band. The separated bands of DNA are cut out and extracted from the gel piece. This step is called elution. The purified DNA fragments are used to form recombinant DNA which can be joined with cloning vectors. So these are the methods. So the diagram is a typical gel electrophoresis showing migrations of undigested lane 1 and digested set of DNA fragments approximately lane 2 to 4. So these are the device and techniques and typical agarose gel structures. Now we start competent host organisms for transformations with recombinant DNA. DNA is a hydrophilic molecule, so it cannot pass through cell membranes. Therefore, the bacteria should be made competent to accept the DNA molecules. Competency is the ability of a cell to make up foreign DNA. The cell is made competent by the following methods. First is chemical methods, and second is physical methods, and third is disarmed pathogen structures. Disarmed structures. Disarmed pathogen vectors. So there are the three methods for transformations with recombinant DNA. First we discuss chemical methods. In this method, the cell is treated with a specific concentration of a divalent cation such as calcium to increase pore size in the cell wall. Calcium increase pore size in cell wall. The cells are incubated with recombinant DNA on ice, which followed by placing them briefly at 42 degrees Celsius and then putting it back on ice. And this is called heat soaked treatments. Second method is known as physical method. Physical methods includes micro injection method, biolistic or gene gun methods. Micro injection methods in which recombinant DNA is directly injected into the nucleus of an animal cell of micropipeters. In biolistic or gene gun methods, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in plants. And disarmed pathogen vectors are also used in this method. Di-armed pathogen vectors are also used to transfer recombinant DNA like Agrobacterium tumefaciensis. So these are the methods by which transformations with recombinant DNA are competent of the host. So now we discuss process of recombinant DNA technology. Process of recombinant DNA technology. Recombinant DNA technology involves the following steps. Recombinant DNA technology involves following steps. First, isolation of DNA. Isolation of DNA. Second, fragmentations of DNA by restriction endonuclease. Fragmentation of DNA by restriction endonuclease. Third, isolation of a desired DNA fragments. Isolation of a desired DNA fragment. Then amplification of the gene of interest then ligation of DNA fragments into a vector. Then insertion of recombinant DNA. Into the host. Then seventh steps, culturing the host cell on a the host cells on a suitable medium at a large scale. Then extraction of desired gene product. And downstream processing of the products as finished product ready for marketing. So these are the various steps by which recombinant DNA technology is formed. So first, discuss one by one of each and every process of DNA. So first we discuss and draw the diagram of the representations of recombinant DNA technology. That is the foreign DNA. Foreign DNA. Inserted in plasmid vectors. Vector DNA. Vector DNA. Vector DNA. Plasmid. Plasmid DNA cut DNA in a specific place. Cut DNA into the place. Then leakages. Then transformation takes place. Transformation into the host cell, equally well of the host cell, DNA equally bacteria, that is the cell divides into two. Two cells. So diagram representations of recombinant DNA technology. Representation of recombinant DNA technology. Ligation of ligases join foreign DNA. Foreign DNA to plasmid. Foreign DNA to plasmid. Plasmid. Ligases. Yes. So these are the structures of recombinant DNA. same restriction enzymes, restriction enzyme cutting both foreign DNA and vector DNA at a specific point, that is a specific points cutting DNA. So these are the structures. Now discuss one by one steps of process of recombinant DNA. First we discuss isolation of genetic materials. To cut DNA with restriction enzymes, DNA should be purified, free from other molecules like macromolecules, proteins, lipids, polysaccharides, etc. The bacterial, plants, animal cell is broken down by enzymes to release DNA along with RNA. Proteins, polysaccharides, and lipids. Bacterial cell is treated with enzyme lysozyme. Plant cells is treated by cellulase, and in fungal cells is treated by the chitinase. So these are the enzymes which is used for different organisms of their cells. RNA is removed by treatments with ribonuclease, and protein are removed by treatments with protease. After several treatments, the purified DNA is precipitated by adding chilled ethanol. Next step, cutting of DNA at specific locations, in which DNA is cut using restriction enzymes. The purified DNA is incubated with the specific restriction enzyme at conditions optimum for the enzyme which they act. Now the process is repeated with vector DNA also. Third steps, isolation of desired fragments. Isolation of desired fragments. By using agarose gel electrophoresis, the activities of restriction enzymes can be checked. Since the DNA is negatively charged, it moves towards the positive electrode or anode, and in the process, DNA fragments separate out based on their size. The desired DNA fragment is eluted out by these steps. Then amplification of gene of interest by using PCR. PCR stands for polymerase chain reactions, in which the polymerase chain reaction is a reaction in which amplification of a specific DNA sequences is carried out in vitro. In vitro conditions, the DNA sequence is carried out. This technique was developed by Carey Mules. Techniques developed by Carey Mules in 1985, and for this he received Nobel Prize for Chemistry in 1993. So in next class we discussed the complete process of amplifications of gene of interest using PCR method. So polymerase chain reactions were discussed in next class. Thank you.